Case Western Reserve University's Institute for the Science of Origins proudly presents Real Science Now, featuring top experts in science and medicine, covering everything from new planets to curing cancer to virtual reality and many topics in between. The Real Science Now lectures are hosted by the Great Lakes Science Center and presented with the assistance of Case Western Reserve University, its College of Arts and Sciences, and MediaVision. Thank you for joining us. My name is Glenn Starkman, Director of the Institute for the Science of Origins. Today's program features 3 presentations entitled Cancer Moonshot, Sea Change in Cancer Research, MRF Imaging, Revolution in Detecting Cancer, MS, and Heart Disease, and Stem Cells, New Strategies Against Brain Cancer. Let's begin with Dr. Stan Gerson talking about the Cancer Moonshot, a program to make major improvements in cancer prevention, detection, and treatment.
I have the distinct pleasure of talking about the Cancer Moonshot and explaining to you what it is. So in the State of the Union, President Obama asked Vice President Biden, looking over his shoulder, to lead a new initiative called the Cancer Moonshot to eliminate cancer as we know it and to shorten the time it took to get through some of the critical inventions to patient benefit, and actually announced a $1 billion initiative. And I just noticed that Congress has supported this with a $600 million request in their fiscal budget for 2017. So what does it mean? Well, technically it's laid out by the Vice President and the President. It was to accelerate progress in cancer discovery, prevention, and screening, break down barriers in patient access to the best treatments, get rid of the silos of databases that we have, get better collaboration between industry, academia, government, so-called public-private partnerships. So those are incredibly lofty goals. Our job is to figure out how to actually do it. He also asked for acceleration of FDA drugs to be able to accomplish in 5 years what would otherwise take us 10. And it allows us to envision no longer experiencing cancer as we know it. Again, incredibly lofty goals. For any of you who have had a family member go through a cancer experience, it isn't good, and it's rare that someone comes through that experience wishing to do it all over again. So we know it's a major disease, and I'll remind you that Vice President Biden's son also went through a glioma experience and died about 16 months after diagnosis. So the Vice President took special sort of haunting interest in this initiative. So I'll just back up for a second and just remind you, what is cancer? Why is it such an important area and an issue? It's a function of abnormal growth of normal cells. They develop a variety of changes that allow them to grow outside of the box, if you will, to take on characteristics that normal cells don't have. And it's something that we've known is critical to evolution since the dawn of time. You have to have mutations, you have to have evolution. The challenge is keeping normal cells in check and abnormal cells dying off. So there are 10 hallmarks of cancer. I won't take you through all of them, but suffice it to say, there are many, many different ways that cancer cells can grow abnormally and can emerge in overwhelming a normal cell population, whether it be on your skin, in the breast, in the brain, in the lung. Some may be activated by carcinogens, smoking, alcohol, some by metabolic changes. Being obese raises the risks of cancer, and some are simply the consequences of age. We know that cancer rates go up. In fact, as we start to live longer, cancer rates accelerate. And we also know that populations are at risk when they change geographic location. It's not the sun and the weather, it's lifestyle and food intake. So we know that when Chinese come to the United States and start taking on the Western diet, they're at higher risk for cancer and the like. So there are a variety of observations around cancer. Cancers have mutations. There's an abnormal growth of cells. These genetic changes allows them to grow more differently. They forget to differentiate and die. Every day your skin falls off and a new layer of skin comes up underneath. A tumor cell on the skin forgets to die, it keeps growing, and that's why you get a mound, that's why you get a melanoma. UV from sun exposure is a perfect example. And then, of course, the cells can leave their primary location, spread elsewhere, and become metastatic. And they also start to utilize metabolic processes that are very different. They eat sugar rather than fat. So that's why there's been an interest in diet restriction in patients with cancer, or even in cancer prevention. We've also noticed that cancer cells seem to act like stem cells and can grow in a variety of ways that are very, very hard to get rid of. We've noted from mutation analysis now that no cancers are the same. So we can look at this in a variety of ways, and we can understand how normal cells can start to mutate, how one mutation may make the cell actually less robust, another mutation make the cell more robust. It's a random process that may take years and years and years. We estimate that going from a polyp to a colon cancer is on the order of about 8 years, which is why we ask you to get a colonoscopy over the age of 50 every 10 years. So how are we going to understand this and get rid of cancer? It takes a huge combination of things, and it's a complex task. There's not one cancer, there's thousands of different types of cancers. Vice President Biden likes to say there's 200, but that's because the histologies are 200. We know from each of those 200 there's 100 underneath it. So there's many, many, many different types of mutations that can cause cancer. It's not as simple as controlling your cholesterol. The men in the room are taking Lipitor. Lipitor cuts down the cholesterol level uniformly because there's one enzyme. You can inhibit that enzyme, you're done. In cancer, it doesn't work that way. One enzyme may be mutant, the other may not work. A third may counteract the inhibition effect of the first. So it's a much, much more complicated task. Now we're playing a critical role because within our catchment area, we see around, it says here 80%, more like about 65% of the population come to either university hospitals or the Cleveland Clinic or one of their disciple organizations, hospitals, outpatient clinics, and the like. And it's an incredible responsibility. There's a greater proportion of cancer patients seen under the umbrella of the Case Comprehensive Cancer Center, one of the 67 NCI designated centers, than any other center in the country. So we raised our hand when Vice President Biden took on this initiative. I spent a lot of time in the Vice President's office in the last 3 months working with him and his team on how to overcome this devastating illness and advising him that Cleveland was a special place to think about some demonstration projects because of our integrated efforts, our coordination across the population, and our outstanding science. So who are we, in fact? So just within our 15-county region, there's almost 4 million people. As you can see, about 23,000 cancers. And we see around 15,000 of those, so about 65%. Now, we have some special issues in this region. We're not happy about them, but they're real. We're fatter than we should be. So if you can see, the national obesity rate's around 28%. And although in Cuyahoga County we're a little bit better than that, in Cleveland we're certainly worse. If you look at cigarette smoking use around the country, it's about 17%. In Cuyahoga County, it's 21%. But that 21% is 36% in the city of Cleveland and more like around 11% in the suburbs. So our affluent suburbs do fine. We don't have to worry about them. But our city is in big trouble, and that's because single cigarette use is rampant. You can buy a single cigarette for $0.25 at the local store. Cleveland has just passed a age 21 rule, up from 18. That may have an effect. We don't know. Over the next few months, we'll find out. And it's a poor city. It's of interest to us that perhaps one of the benefits of being recognized by winning the Cavaliers championship, that maybe the nation will take a look at the poverty rate in Cleveland, which is much too high, with about 18% of our population in the county below the poverty line, which is, again, well above national averages. One additional item that we're concerned with more recently is HPV vaccination. It used to be that we would think about HPV vaccination as a variable. Have it if you want, maybe if you don't want. But we now recognize that HPV is the solo way to assure our children that they won't be at risk for a variety of cancers, whether it be cervical, head and neck cancers, and the like. Yet our HPV vaccination by the age of 13 is only 11%. In some areas where it's been mandated, it's up to 40 or 50%. It should be 80 or 90%. So we have a socioeconomic problem, which is linked because of our risk factors to the scientific issues. Some people might think poverty is poverty. We think poverty is a scientific question. We have too many people who have a diagnosis of a life-threatening cancer, and we know that we have many, many underserved populations. We can prevent this by reducing our smoking rates, increasing HPV vaccination, encouraging a healthy lifestyle. So our problem is to move ahead with cancer at every stage. So when we went back to the Vice President, we suggested, in addition to what he had promoted, to focus on adolescent and young adult cancers, to worry about cancers which he was well aware were bad cancers, and to look hard where we need to develop new approaches. The reason we're interested in the technologies and capabilities that we have now, and you'll hear from Mark Griswold next, is that we have amazing technologies in imaging, and I'll let him describe it, including imaging of brain and brain tumors. We have incredible opportunities, you'll hear from Kara Zhang, in understanding the linkages between genetic abnormalities and our drug development efforts. And we are active in helping the Vice President figure out ways to be able to use large data sets, drug evaluations by the FDA, to improve and make more quick the transition from a discovery in the laboratory to a benefit to a patient. So the timeline is actually pretty short because this is the last year of this presidential era. So the State of the Union was in January. We actually were forward enough to be developing some op-eds in Crane's Cleveland Business and in the Plain Dealer. In February, we submitted some suggestions in the Vice President's office in March. That led to 3 invited meetings at the Executive Office Suite in Washington. And then we had Vice President Obama visit here at the end of June. In addition, we got some notoriety because we've been part of the national discussion. Alex Wang at our institution is part of the Blue Ribbon panel for the National Cancer Institute. I've been invited and participated in the National Summit. And we had an Idea Stream supported event where we actually talked about these issues on NPR and WVIZ on the day of the National Summit here. So the week also allowed us to introduce Monica Hooper, who you see in the upper right, who's our new director of the Office for Disparities Research. And she is helping us move forward to make sure that our underserved populations are taken care of. We got the attention of Idea Stream and Ideas, and as I mentioned, we brought Vice President Biden to one of our community locations to help him underscore the importance of HPV vaccination and smoking cessation. So what's happening in the national level? So on the national level, there's been a lot of talk. There's a federal task force, NCI committees, Blue Ribbon panels, but by October of this year, before the next year's budget gets implemented, the NCI will put forward proposals to spend an additional $600 million specifically on the NCI Moonshot research programs in all these areas that I've described to you, whether it be drug discovery, genomics, outreach, and community benefit. We're hoping, this isn't dollars coming here, this just happens to be a picture I took when I was on the top floor of the Executive Office building, which is now the Cancer Moonshot location. What's of interest is that there's been over $1 billion identified of additional funds in public-private partnerships We are looking hard for those funds to come to Cleveland, but they are helping out immensely in drug development and in immunotherapy. And we're hoping that that leads to further engagements across the NCI, the VA, and multiple cancer centers. So as we move forward, you will hear some of our other scientists today. You heard about from Jeremy Rich earlier, as well as John Karn. You'll be hearing from Mark and Kara Zhang. These are the scientific areas which are being pursued, all of which are viewed as critically important and ones where we can make significant progress just in the next few years. We, of course, want to be central to the data sharing element of this. It's amazing how much of our data is put away in electronic medical records in individual siloed databases. Too much data is being left on the table that individuals cannot utilize. So we are participating. We're the largest cancer center in the country right now, participating in a national effort called CancerLink, where patients' information will be anonymized and then downloaded. And we're developing through our hospital systems, including at Metro Health Medical Center, a ClearPath program that will help us move forward to share data in the city of Cleveland. In addition, the one program that we're most excited about is implementing a real effort to cut down the smoking rate in the city of Cleveland, currently around 35 or 36%. We'd like to get it down to 22%. Very recently, I was in City Hall talking about maneuvers that we can do with the City Board of Health. We've talking actively with the County Board of Health to do that as well. And we want to increase Hpv vaccination rates. Those rates should be up around 70 or 80%. They're currently at 11%. So we're hoping to get this accomplished. What caught the Vice President's interest is that we're combining our efforts with the District of Columbia, who also have a concentrated underserved population with the same issues of a high smoking rate and a low Hpv vaccination rate. So we will continue to move through these issues and link together as well as we possibly can issues around data sharing, prevention, and decreasing barriers of implementation. So we will take advantage of funds as they come forward. As you can imagine with projects like this, there's a huge amount of sweat equity, but Cleveland's the right place to do it. Thank you.
I was just curious as to, do you have the percentages of cancer patients that are actually also smokers? Because I know that you're promoting this 36% in the Cleveland area, but the 3 people that I know that are adults that have cancer right now are not smokers, and then I think about all the children. So I guess I was just curious as to, as far as the population of people that have cancer, how many of that percentage were actually smokers?
So of people with cancer, what proportion smoke? So we're not 100% sure about that. I'm pretty sure that the data is tracking so that, again, in a suburban population, it's much lower than the urban population. What we do know is that urban smokers who develop cancer, it's much harder to get them to quit than the suburban smoker who develops cancer. So we're pretty sure that those numbers are pretty parallel to this, on the order of 30-35% in an underserved population, and around 15 to 17% in a suburban population. So they tend to track, and we are getting our major hospital systems to work hard at identifying those individuals with cancer who smoke and be very rigorous in smoking cessation efforts.
You've been watching Stan Gerson talking about the Cancer Moonshot. Up next is Dr. Mark Griswold discussing improvements in MRI technology that should revolutionize diagnostic medicine.
As Stan mentioned, I'm going to talk about imaging, but I'm going to start with some maybe obvious observations about the weather. So last year I had to go to Brazil in the middle of winter, and this was the weather the day that I left. So it was about -20 here, and it was about 100 in Rio, where I was going. And so we could say that it was cold in Cleveland and hot in Rio. You might say very cold in Cleveland and very hot in Rio, depending on where you are. But if we ask this person, he may say, well, actually, that's pretty hot in Cleveland compared to where I normally live. We can also look, and there's good data that shows that the Earth is warming from year to year. But it's very difficult to recognize that. So if I asked you what the temperature was on this day last year, you would probably have no idea what that was from your memory. You could look it up in a database. But that's a very difficult question to answer. And so just to summarize that, we use words to describe situations, but different people use different words to describe things depending on their experiences. So that description is not reproducible. We're also not sensitive to global changes, and that could lead us to big problems. So this may be obvious, but this is why we use numbers to describe situations, because numbers allow you to make good decisions like what to wear today, and your subjective feelings don't. So if I look out the window, it may be sunny, but that doesn't mean that it's warm enough to wear shorts. Now, what does this have to do with imaging? Well, if we look at the field of MRI that I work in, it's a magical piece of technology, but it's completely qualitative up till now. So the way MRI works is that a radiologist would look at this spot and they would say effectively, this spot's too bright. And that may relate to a disease, but not directly. So if you look at the image, actually the brightest thing in that image is the fat around the skull. So now, as I said, numbers would allow us to make a better decision here, but it's really difficult in MRI. And I'm going to try to describe to you why it's difficult in a very few minutes. We would normally teach this over 2 semesters, but we're going to try it in about 2 minutes. So MRI, in my mind, is magical. It's the most unbelievable technology that has been invented in my lifetime. It's based on the quantum mechanical property of spin that exists in protons in nuclei of hydrogen atoms. It's completely magical. You take a bunch of water in this case, and you put them in a big magnetic field, and each one of those protons inside those hydrogen atoms will start to look like a little bar magnet. And it's only about 1 in a million of them that are pointing in the right direction, but they basically add up to be a little bar magnet that we could then manipulate. What happens is that we can use another magnet to distort that magnetic field in the protons away from where they're normally aligned, and then they wiggle back to where they were. And that process of wiggling back up is called relaxation, and that's actually the thing that we care about, is the time that it takes for that process of going back to the way it was to happen. So there's 2 numbers that I want you to pay attention to. One is the time that it takes for the magnetization to regrow along the direction that it was initially pointing. We call that T1. And then if it's disturbed away, there's a component that's perpendicular to that, and the time that it takes for that to disappear, we call T2. So what we do is, in MRI today, is that we'll do the same thing over and over again to build up some information about one of these 2 numbers, and that will become the image that we'll use. And these are important because these 2 components actually have to do with molecular level structures of tissues. So T1 in particular relates to the number of proteins and membranes that are in a cell, so you can imagine that this has changed quite a bit when we look at cancer. The other thing is T2 tells us about how freely that water can move around. So you can imagine that if it's in a big, thick, gooey fluid, it would move differently than in free water. And so these 2 numbers are effectively molecular level probes of tissue. And so it would be great if we could quantify these numbers, because they should tell us exactly what's going on inside the tissue. The problem is that these are times that I talked about, and the curves that define them are somewhat complicated exponential curves. And so if we were going to collect the quantitative information here, we would need to get 5 or 10 images of the T1 side and 5 or 10 of T2. And what that means is that the total scan time of our MRI would go up by a factor of 5 or 10. And anybody who's had an MRI already knows that they're long enough and painful enough so that that's not ever going to happen. So what we do is we live with just one of these from each side, which we call a weighted image, which gives us an idea about the number T1 or the number T2, but they're not a quantitative result. And this works really well, but it leaves us with some big problems. So if somebody comes in with a lesion like this, we don't know whether this is a metastasis, which is a cancer which comes from somewhere else in the body, or whether this is a primary brain tumor that started in the brain. And obviously those have two different treatment pathways, and it would be very nice to know that going in. But the way this is today, we don't know, we can't tell the difference, and so we have to do a biopsy. And just so we're on the same page about what a brain biopsy means, this means that you've got to go in the OR, you've got to have an anesthesiologist and a neurosurgeon. They're going to drill a hole in your head to get a needle in, and because you've got a hole in your head, you're going to spend a day in the ICU. And this is all just to figure out what to do next. We're not even doing any treatment at this point. The cost in this procedure is $12,000 to $51,000, again, just to decide what to do. I would argue that this has got to change. This is outside of the way we want to practice medicine. If you think about the medicine on Star Trek, which should be our future, they would never, ever think about doing something like this. We should aspire for something different. So we did, and I'm going to skip about a decade of research, but what we have come upon is this idea called magnetic resonance fingerprinting, or MRF. And at its basis is a removal of this concept that we're going to do the same thing over and over again. Actually, what we are proposing to do is to randomize the entire acquisition that we do in MRI. What we see when we do this is that the response from each different tissue of the body is different. It's completely chaotic and random, but we can predict it exactly from quantum mechanics, which then means that we can feed this to a pattern recognition algorithm, which then can tell us about the tissues that the information about the tissues that we care about, the T1, the T2, the cellularity, and things like that. And we called it fingerprinting because it's a direct analogy to conventional fingerprinting that the FBI would do. So if we look at the image of a fingerprint, it's essentially a random series of black and white lines. It doesn't tell us anything by itself. We get the information that we care about by matching it to a database, and then we get the person's name, address, and so on. It's the same thing in MR fingerprinting. We have a random signal evolution that we don't know really, just by looking at it, what it is. But we can use quantum mechanics, again, to exactly calculate everything that we would expect to see. Then we can use a computer pattern matching algorithm to figure out exactly what this is, and then we can understand that was a tumor at this position, that was muscle, and so on. So where conventional MRI gives you these qualitative maps, which again are very powerful, fingerprinting gives us a multi-dimensional readout of these times. You can think about them as temperatures. And once we have that multi-dimensional readout, we can then start graphing them and actually start collecting data in populations. And while that one dot on that graph may not be useful itself, when we start doing this across a population of patients, what we see is that different diseases start to cluster into different groups. And so now I know that that tumor that's there is a glioblastoma, which is a very aggressive form of cancer, just based on this MRI exam. I don't need to go do a biopsy. We've shown this now in prostate cancer. So this is a three-dimensional readout of a fingerprint, and you can see the difference. It's hard to read here, but the upper bars are on all three parameters are normal tissue. The lower ones are cancer. You can see in each of these different measurements, we have a quantitative difference between normal tissue and cancer. We can then take those three numbers, those three parameters that differentiate this tissue, and actually make a score. So everything to the right is normal tissue. We don't need to biopsy that tissue. Everything to the left has some sort of abnormality, and we may need to biopsy that. So, and we think that we can even reduce the ones on the left even further. But this alone, this one imaging experiment could reduce the number of biopsies of prostate by half or so. That's a big change in our mind. We're also able to start to look at the brain and to see changes that are just due to aging. So this is a set of nominally normal people, and we can see differences across different regions of the brain that change with age. So if we now start studying disease populations, people with Parkinson's, Alzheimer's, MS, we would expect their numbers to deviate from these nominally normal people, and we can probably see that before it's apparent clinically. And if that's true, then we can start even to treat it before it's clinically relevant. We're even, unfortunately for the men in the room, able to see differences in neurodegeneration between men and women. And unfortunately, it's accelerated in the men compared to the women. So just, sorry guys. In MS, we're able to see changes in otherwise normal appearing tissues that are correlated to the disease and correlated to clinical outcomes. So that NAWM is normal appearing white matter, and we can see that there's differences there in MS compared to healthy controls. And this gives us a window to initiate pharmacologic treatments of these people before their lesions have actually damaged their brain to the point where it's not recoverable. I'm not going to show you disease today, but we can also do this in the heart to start studying the structure of the heart and other diseases of the heart, again, before they become an issue. There's about 49 groups that have picked this up around the world, but I want to point out Stanford. They have developed an alternative method here that allows them to solve for parameters of tissue like the average capillary size, the cerebral blood volume that's there, and the amount of oxygenated blood that's getting to that tissue. So you can imagine in tumors and in stroke, these are going to be critical parameters that are going to tell you about the physiology of that tissue. And I don't need to go get a biopsy to understand that. The last thing I want to do is biopsy the brain. And now I can do that just by imaging. So last but not least, I want to go back to this idea of MRI. Anybody who's had one understands that it's a little bit uncomfortable. It's very loud and confined space. And we are thinking about ways to make this more comfortable for the patient. So if we go back and we think about this process, what I said very quickly was that in fingerprinting, we randomize everything. And that's the only requirement for this method, is that it's random. The cool thing is that there are a lot of things that are almost random in our world. And one of my favorites happens to be music. In fact, this piece is one of my favorites. So this is Yo-Yo Ma playing Bach Cello Suite 1. I hope everybody can recognize this. But what we can do is take this as a starting point and then design our entire acquisition around this piece of music. And so what this then becomes is a scan that looks like this. So we're going to start again with Yo-Yo Ma and then transition into what it sounds like in the scan room. So no more banging, no more screeching MRI scans. You can go listen to Bach, hopefully. And because it's just a different form of random, actually the efficiency and the quality of the results we get from that are the same that we get from any other random solution that we do. So this is one particular form of random that I really like. So to sum this up, we think that we are switching to now having an MRI method that gives us numbers, and that those numbers mean something, and that we hope that this will lead to better patient care. We're developing this in Cleveland and sending it out to the world, and I think that we're going to have some great success coming forward. I just want to, before I get off the stage, quickly thank the big group of people that actually do the work here, just to show their pictures. Thank you to everybody.
With your fingerprinting technique, is it possible to take that data and explore it with the different algorithms afterwards, like exploring with the way Stanford does it, or a different way after the fact, in order to explore what you had way in the future? You might think of something later.
One of the things that we're trying to get ready for is the big data revolution that's coming in our world. If you go to Google and you look at, you can go to Google and search for images like this one. or you can search for images of kites or dogs or whatever, and it knows these things by now. We're on the edge of being able to do this in medical imaging as well. And one of the key things that we have to have ready is a true quantitative readout like what we're doing. So we're hoping, we have many more analyses that I didn't talk about today about the texture analysis, how one area of a tumor relates to another that we think will relate to cancer. We're able to look inside a single pixel of that image and understand the microstructural changes that are happening there. And then we're hoping that this whole big data machine learning thing kicks in as well and will enable us to do analyses that we can't even imagine today. But again, we have to come back to have this quantitative result that's reproducible from day to day to day before we can think about that. So that's why we spent a decade on developing this. That's a great question that comes directly from this. So.
You've been watching Mark Griswold talking about magnetic resonance fingerprinting and its huge potential for diagnostic medicine. Up next is Dr. Jeremy Rich discussing how stem cells could be used to treat brain cancer. Now, back to the talk.
I appreciate the opportunity to present some of the concepts that are really taking on new light in the area of stem cell biology linking to cancer. So I'm a neuro-oncologist. That means I treat patients with brain tumors. The most common intrinsic brain tumor is glioblastoma. This is a disease that is universally lethal. It is what killed Ted Kennedy. And we can see pictures here of both an MRI scan as well as the unfortunate outcome of a pathology of glioblastoma. So many years of research have therefore transformed our therapy. We now integrate upfront the use of an oral chemotherapy agent called temozolomide. This was a raging success. As you can see here, these red and blue lines are shockingly different. I'm being sarcastic here. So this is really something, though, that we have seen only a 2.8 month increased survival, and yet we spend now probably $1 billion a year on treatment with this chemotherapy agent. This is really suggesting we have a great deal of requirement to improve things. In fact, you can see this where if you look at a 5-year annual survival of some major cancers, you can see 90% of some individuals in common cancers, for example, in children with leukemia or in breast cancer patients, will survive that 5-year rate. But it's substantially less than 10% within the context, even in children where brain tumors are actually the most common cause of childhood cancer deaths. And this is just kind of a shocking output. So this is actually the change in overall survival of patients with cancer over the last 30 years. And you can see 30 years of effort, and we have changed brain tumor outcomes by only 2%. So a really shockingly poor outcome that we really need to change. In fact, brain cancer, if we do a quantification of the overall cost of cancers, brain cancer comes out number 1. So we spend the most and we do the least. So I'm a highly successful clinician, as you can see. Interestingly, brain tumors, particularly in the context of gliomas, this is, again, the most common type of brain tumors, have a very strong racial and gender bias. So it's interesting that they affect minorities to a much lower degree than whites, and we don't understand why that is. More interestingly, in terms of some biology, it turns out that males are about 1.6-fold more likely to have gliomas than women. And we actually are starting to understand some of that biology. So it's quite a fascinating area. But when we really want to make changes, we start thinking about cells. And the cells that are most attractive are stem cells. And we have to ask ourselves, with all the hype of stem cells, is it the wave of the future, or is it snake oil that we're trying to sell? And so stem cells and brain cancer actually are touching on a connection in 3 different areas. One I won't touch on, but that is that there's increasing evidence that at least a subset of brain cancers start off with a stem cell gone bad. So that is the initial event that occurs to start that cancer is actually occurring very commonly in stem cells. The next possibility, which I'll touch on just very briefly, is the idea that stem cells of different types could be used to treat brain cancer. And finally, the area that I spend my most time on are the stem cells that are being co-opted, acting in a very malignant fashion within brain tumors? And the answer is yes, and I'll show you evidence to that regard. So I thought I would take a step back and actually touch base on one of the favorite areas I like to talk about, and that is going over stem cells. The real issue with stem cells is actually there's a lot of confusion in the popular press, and most of the time, because things are complicated, we tend to reduce the complexity when we discuss these issues. And so it turns out stem cells are a very rich area, some of which may be controversial, but most of which is actually something that each of us in the room are dealing with on a day-to-day basis. So the first thing is, what is actually a stem cell? Well, stem cell is defined functionally. It is a cell that has the capacity to self-renew, that is, create a copy of itself, but also create a differentiated cell. So it turns out a stem cell by itself actually really does nothing. So it's kind of like a kid going to college. My daughter's starting Stanford in the fall, and so hopefully she'll do something useful, but they end up initially undifferentiated, and so they have a lot of potential, but they can't really do anything. But then over time what happens is there's a differentiation, there's a skill set that's being acquired, and this is the thing that a stem cell does. It has those dual abilities. And so this is actually something that we can see is present in a lot of tissues, and that is you start with a cell that continuously, as time goes on, has the ability to maintain itself, and that is, you can see that on the left-hand side there, you see that there's a manifestation of self-renewal, that is the ability to maintain itself, but then it also populates the tissue. It has the ability to grow the tissue from which it's derived. So where are stem cells found? And the fact is that what you see in the top there is a fertilized egg, and that is a stem cell. That is the one cell we can point to in a dish and say that is a stem cell. But over time, as one cell becomes two and two become four, and we eventually create what are called embryonic stem cells that are a part of a blastocyst, this is where actually most people pay attention to, and I'll touch on that. But the reality is that as we grow into a fully developed person, we actually have tissue-specific stem cells throughout our body. In fact, we would die without these. So first off, embryonic stem cells. So where do we find them? So they are a very restricted population in blastocysts, and actually they last only, frankly, about 24 or 48 hours by themselves. So when we're using them in a dish, it's actually something that is an artificial situation. And by the way, I don't actually do embryonic stem cell research myself, but so embryonic stem cells can be taken here and maintained. And so they are actually very special because they can create any cell in the body. All right, so that is something we call pluripotent. They can do anything they possibly want. So they can make muscle cells, nerve cells, lots of different kinds of cells. So we call that, again, pluripotent. Tissue stem cells, on the other hand, are things that are found in each tissue in our body. There are some tissues where we haven't been able to figure out exactly which cells are the stem cells, or there are some differences. Actually, it turns out most tissues have at least two different areas of stem cells or two different populations, which makes sense because if one goes wrong, you want to have a backup. But you can see in almost every tissue in the body, we've been able to figure out stem cells. And so stem cells have the ability to create all the tissues in that organ. So we call that multipotent. So they can't generally become tissue types from which they weren't derived. They are more restricted. So my favorite tissue, which we already heard about, is the brain. It is the only important organ to study. And that is that a neural stem cell has the capacity to become three different major types of cells. You see neurons. These are the nerve cells that we really focus our energy on. But actually, they're the wimpy guys who are a small fraction of the overall population. You have the astrocytes, are named because they can be star-like in morphology. And then you have the oligodendroglial cells. And those are the cells that actually act to insulate the nerve cells so that they can function better. More recently, we have something called induced pluripotent stem cells. So this just received a Nobel Prize a few years ago, and that is the idea that you can take a cell and you can reprogram it. You can take a more differentiated cell and make it a more embryonic stem cell-like. Now, it's not entirely the same as an embryonic stem cell. They have some still manifestations of the cells from which they're derived, but these cells have the ability to become any cell type in the body, and so it's a very exciting situation. Let me give you one example. And this is something that we're trying to figure out if we can avoid using embryos. And so one of the cool things, this is a paper that was published back in 2007, where they took a mouse that had a model of sickle cell disease. So those of you who don't know, sickle cell disease is caused by a single point mutation in the hemoglobin gene. And so what ends up happening is there's a protein that's dysfunctional. And this is a lifelong challenge for the patients. They require a lot of care, are likely to have infections and strokes and lots of other problems. So they took this mouse, they took the cell's type of interest, that is the hematopoietic stem cell, they actually induced pluripotency, corrected the problem, and then gave the mouse back a transplant essentially of their own cells and cured the mouse. So this is a really cool thing. So we're now using this to actually ask a lot of different questions about a lot of different diseases in ways we were never able to before. Well, now we can apply stem cells to brain cancer. So one of the things you can do is you can actually take a cell population that's stem-like. And it turns out that brain cancer cells put out evil humors. We have fancy names for these, cytokines and chemokines. And those attract these stem cells. And these stem cells will go and track these cancer cells. And you can use them to actually cause the cancers to grow more slowly. So this is just one example. We can take two different types of cells, one a hematopoietic stem cell, the other a mesenchymal stem cell. And you can treat, and you can see on the right there, these are survival curves. And the further to the right you get, the more survival there is. And so there's the neural stem cells are actually better than another cell type in terms of prolonging survival. And so now there's a clinical trial, or actually a series of these, taking neural stem cells and doing something pretty cool with them. So if these cells can go track these cancer cells, the idea is to say, all right, let's use them as delivery agents. And so you can give genes that will end up causing the local cells to die. And so this is actually something that has been used in clinical trial, some preliminary data that suggests that it's a promising first start. So it suggests stem cells can be used for the treatment of brain cancer. But we also know that there's some bad things with cancer. So this is an Economist article a few years ago. They referenced some of our work, and that is to try and say, can we really make a fundamental change in cancer? Because if we think about it, patients who have advanced cancers, if you were diagnosed in 1971 when Nixon declared the war on cancer, the likelihood of you surviving, that is absolute, never mind duration, but actually survival, is not much different 1971 to now. And that's kind of a shocking idea. The way we've improved things is we have earlier detection, we decreased the toxicity, but ultimately if you have metastatic cancer or lethal cancer types, the likelihood you will survive in the long term is almost the same as it was more than 40 years ago. Well, why could this be? Well, it turns out these things called cancer stem cells I'll talk about momentarily have the ability, like normal stem cells, to self-renew, that is, make copies of themselves. They make the tissue from which they're derived, tumors, and they are therapeutically resistant. And actually my group has helped sort of define some of this. So you can think about this as using a lawnmower. You can go and you can remove all of the plants growing above ground, but what ends up happening is you have the roots. And we really think about cancer stem cells as kind of the roots there that will allow the tumor to regrow. So we have helped in defining elements of what we call a cancer stem cell. I will be honest with you, it is a controversial area. But we know across the top here, we can see that they're absolutely defining characteristics, again, based on the relationship with normal stem cells. And that is the ability to self-renew, the ability to make a tumor. There are lots of other aspects that are unresolved along the bottom, that is how frequent they are. We don't know if your tumor has more stem cells or fewer stem cells, really how much difference that's going to make in the likelihood that you're going to respond to therapy. That's still something we're working on. But we were able to, some years ago, show for the first time that cancer stem cells are preferentially resistant to the most effective therapy for brain tumors, and that is radiation. And later on, or actually about the same time, we also were able to note that a angiogenic aspect, so you heard about angiogenesis momentarily ago, the growth of new blood vessels. It turns out that cancer stem cells have the ability to preferentially grow new blood vessels. And you can see that here across the bottom of those brains. Those are all tumor-bearing brains, but they're much redder in the case of the cancer stem cells. And that's because they have the ability to grow blood vessels more effectively. And we were able to give a therapy called bevacizumab, also known as Avastin, that could selectively block the growth of tumors that are derived from cancer stem cells, but they were really ineffective against the non-stem cells. And this is just not a laboratory issue. Again, as a clinician, I was involved in the implementation of bevacizumab to the treatment of brain tumor patients and led to the FDA approval of this therapy. But ultimately, what we're starting to see is that there's failure of this drug, and we think in part it's because it's causing changes in the cancer stem cells. So I have tried to get the field, and hopefully someday they'll listen to me, but like most intelligent people, they don't, to really think about cancer stem cells in the ways that it kills patients. And so it turns out, shockingly, it's not whether a cell can just grow copies of itself that really determines whether you live or die from cancer. It's metastases, invasion, and the immune effects that, by and large, are causing more of an issue. And further, there's another aspect that's very interesting, and that is a relationship to where they live. So these are kind of the real estate agents of cancer. So it turns out that the location is key. We call this the niche. And it actually turns out that many of our therapies, including radiation and chemotherapy, may be acting more on disrupting that place that they live, disrupting that niche, than actually killing the cancer cells. So just to bring this to an analogy that might be easy to understand, so if we have a set of bees here, and we have a lot of fly swatters, then we can call these either chemotherapy or radiation. We can get rid of those bees, but it turns out that queen is left behind, and that queen can then repopulate the hive, okay? And so we can take this and hopefully get rid of the queen and get rid of the entire hive. So we want to do the same thing with cancer. So we use radiation, chemotherapy, zap, zap, zap. Unfortunately, chemotherapy and radiation both leave behind that cancer stem cell, and it can regrow the tumor. So what we're starting to do now is ask, how are these cancer stem cells regulated? And you've heard some elements of normal biology, and it turns out, for example, we recently showed that if you have lower levels of sugar, that glucose, turns out cancer stem cells like that, and they're more likely to survive. We have also seen that those parasites that you heard about, those are the cells that line blood vessels. It turns out that cancer stem cells can become parasites. So there's a lot of interesting interactions with normal biology and cancer biology. But one thing we're trying to do, again, as a clinician, is find new therapies. So this is just one drug that we published last year that we're starting to see that if we target cancer stem cells, we can have an impact on outcome. One of the challenges is cancer stem cells are not uniformly distributed. That is, that they live in different locations. Low oxygen levels is one place, and then areas around blood vessels are another place. And so we're starting to use novel approaches, and this is something I've done with one of my former trainees, Gerson, who's now a faculty member at Cleveland Clinic as well. And we're starting to use mice with advanced imaging techniques. And we're able to see that a single tumor cell, you see in the top left here, can give rise to an entire tumor. It's shown here in green. And the shocking thing is, even though we have small amounts of tumor cells in green here that are the cancer stem cells, in just the span of 3 days you see an explosive growth, whereas the blue cells are the cells that are the majority of the tumor cells, and they are not growing very aggressively. Further, we're starting to see that there's a non-random growth pattern around blood vessels, for example. So this is all great, but we're not going to be able to show how the tumors are growing in all the mice. And one of the things we'd like to do is, with the understanding that these cells have got to grow in a certain location, you can't just say, I'm going to put cells in a dish and expect to have representation for patients. We want to get to personalized or precision medicine, and that is we want to take every patient, derive a model from them, and test therapy. So how do we do that? Well, there's something called organoids, and so what we do is we create organoids, and what we do very quickly is to be able to grow these organoids and show that they grow just like the original tumor in mice. And for the sake of time, I'll just show you that we can see that there are two different regions, that they're very similar to normal brains. And we can actually take different regions, grow organoids from those different regions, and they have different biology. And so just at the end, what we can see here is this is actually, we're growing a whole bunch of organoids. We're segregating these different locations, and we can do tests on these different inside and outside environments. And so just at the end, we're creating these look like those little candies that you can get on a wax paper. So we're growing organoids by the hundreds, if not thousands. This is not bubble tea we wanted to show, but rather organoids in general. And so we're using this for drug discovery. So in conclusion, what we're able to say is that cancer stem cells are the therapeutically resistant population by and large. But we can't just target them because it turns out there's plasticity. We have to target with conventional therapy as well. And so hopefully, hardworking folks like these folks here will help. But ultimately, this is something that's really a partnership, not just with researchers, not just with industry, but the patients themselves. So thank you very much.
The Real Science Now lectures are hosted by the Great Lakes Science Center and presented by the Institute for the Science of Origins with the assistance of Case Western Reserve University, its College of Arts and Sciences, and MediaVision. For more information on Real Science Now and the Origins Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.